The story of cell death, from chaos to coordination. Once upon a time in the early days of cell biology, death inside the cell was seen as nothing more than a tragic accident, a chaotic, unregulated collapse triggered by injury or stress. Like an old machine that had simply worn out or short-circuited, a dying cell was assumed to fall apart haphazardly, with no guiding hand, no internal script. Pathologists labeled this process necrosis, a messy, uncontrolled unraveling, leaking cell contents into the surrounding tissue, often leaving inflammation in its wake. But science, like life, is full of surprises. This simplistic view began to shift dramatically when researchers uncovered something extraordinary, a different kind of death. Unlike the noisy demise of necrosis, this new form was clean, quiet, and most surprisingly, regulated. It was called apoptosis, and it changed everything. Unlike necrosis, apoptosis was not the result of overwhelming damage, but rather an elegant, internally scripted process. The cell didn't simply break down, it followed instructions. It condensed, shrank, fragmented, and wrapped itself into neat little packages which were then safely removed by phagocytes. No inflammation, no mess, just a graceful exit. This was not death by accident, but death by design. Even earlier clues had whispered that cell death might not always be so chaotic. During the development of organisms, from tiny invertebrates to complex vertebrates, scientists noticed cells vanishing at precisely the right times and places. Could death itself be written into the blueprint of life? In the roundworm Chinorhabditis elegans, this became strikingly clear. In every hermaphrodite embryo, exactly 131 of the 1,090 somatic cells die at predictable stages and positions. In males, 147 out of 1,178 are fated for death. These were not coincidences, they were instructions, a plan, a program. This led to the birth of a new concept, programmed cell death, PCD, a term coined to describe this deliberate pruning of cells during development. As researchers, including the legendary Robert Horvitz and his team later discovered, this process was driven by a genetically encoded network, a cell-autonomous signaling system that orchestrated who lived and who died. The curtain had lifted on a new biological drama, and death was no longer a villain. It was a sculptor, carving form and function into living tissue. But the story didn't end there. As we began to map out the roles of PCD beyond development, its importance only grew. In adult tissues, regulated cell death, RCD, became essential for maintaining balance, or homeostasis. Old or damaged cells had to be removed to make room for new ones. Immune cells, after clearing an infection, had to step aside through apoptosis to prevent chaos. And when infected or cancerous cells threatened the body's integrity, death became a line of defense. And yet, just as there isn't only one way to live, there isn't only one way to die. Over time, scientists identified multiple forms of regulated cell death. Apoptosis was just the beginning. They discovered necroptosis, a controlled but inflammatory death, more like necrosis in its dramatic flair but still genetically regulated. Then came pyroptosis, a fiery, immune-triggering death commonly seen during infections. These forms didn't just remove cells, they announced their departure, unleashing internal contents that alerted the immune system. Molecules like DNA, ATP, and metabolites poured out, acting as danger signals and rallying immune defenses. Each type of death played its own role in the grand biological narrative. Apoptosis, the silent surgical executioner. Necroptosis, the alarm bell. Pyroptosis, the fiery sentinel. And now, another intriguing character has entered the stage, autophagy. Autophagy, meaning self-eating, began as a mechanism for survival, cells consuming their own components to endure stress or starvation. But under certain conditions, it can tip into a form of self-destruction. Unlike the others, autophagy blurs the line between life and death, resilience and surrender. In disease contexts, including cancer and neurodegeneration, it adds a new layer of complexity to the tale of cellular demise. This evolving understanding of cell death has not only deepened our knowledge of biology, it has opened new doors in medicine. From targeting cancer cells that resist apoptosis to modulating inflammatory forms of death in autoimmune disorders and infections, therapies inspired by the mechanisms of RCD are now in the spotlight. In the end, the story of cell death is not just about endings. It is about balance, renewal, and the intricate choreography that keeps life itself in motion. What was once thought to be a chaotic collapse is now understood as one of nature's most elegant and essential performances.